Now,
I clicked on go live. I'm on the other screen,
yeah.
so I can check when it goes live at least. It should go live now. Yeah,
Now,
I clicked on go live. I'm on the other
Ranav, your camera is not on, so if we
Yeah,
can just
I know, I
begin.
know.
Yeah.
There.
Do you want me to start?
Um, I think everything's work. No, it's not
Yeah, we are live now.
live.
We
See,
can,
there.
yeah.
I changed it. It's now presenting our meet screen. It's working.
All right, so I'll share my screen. And
Yeah, I think we have an intro and then you can start.
okay, that sounds good.
Hello, everybody, and welcome to the last episode of Quintessential Conversations. Today, I would like to introduce Dr. Prarthana Shankar. She has recently finished her PhD in toxicology from Oregon State University. Her dissertation is titled The Mechanism of Actions by which Polycyclic Aromatic Hydrocarbons, or PAHs, cause developmental toxicity. Previously, Dr. Shankar was a student employed at South California Coastal Water Research Project, and now she has taken a full-time job at the U.S. Environmental Protection Agency. Today, she will present her thoughts on environmental and molecular toxicology. Over to you, Dr. Shankar.
Thank you, Ranav. Okay, so you can hear me. Great. Thank you, Rana, for that really nice introduction. It's always fun to do things like this. I really enjoy talking to people about the kinds of research that I've gotten a chance to do. And so today I titled my presentation Environmental Toxicology in the 21st Century. And you'll sort of see as this presentation goes on why exactly I'm calling it that. Rana gave a really nice introduction, which I didn't realize. So I kind of just put here on my first slide, and I'll just run through this quickly. A little bit on my academic journey. So I'm actually originally from southern India, the city of Chennai. I graduated from high school, from specifically AMA Matriculation Higher Secondary School, a small state school in Chennai. After graduation, I had this really nice opportunity to go to California for my undergraduate education. And so I went to California State University in Fullerton. And here I picked up a Bachelor of Science in Molecular Biology and graduated in 2016. As Ranav mentioned, I then decided to go to graduate school. And so I chose Oregon State University, and specifically the Environmental and Molecular Toxicology program there. And just recently defended my thesis in February of this year. And then I just actually, it's been 1.5 months, but started a position remotely at the US EPA, which is the Environmental Protection Agency. And this is a postdoctoral position. And for those not familiar with postdocs in general, this is typically a 1 to 2 year training program that people with PhDs do after graduation to kind of pick up on skills that they might have not gotten during graduate school, or maybe just explore other fields to kind of figure out what exactly they're interested in. And so typically you'll see people doing 1 or 2 postdocs before they then move on to a permanent position. So that's my story. Hopefully this helps sort of understand the presentation a little bit better. I thought I'd start off with kind of very broadly just looking at this word pollution. And National Geographic defines pollution as the introduction of harmful materials into the environment. These harmful materials are called pollutants. I wanted to sort of highlight that pollutants can be both natural or man-made. And so in the figure on the left-hand side, we're looking at a volcanic eruption, and this is absolutely a natural phenomenon. And what you're seeing here is a bunch of ash that's being produced from this volcano. And just because it's natural, it doesn't mean that it's good for us. There's obviously tons of toxic chemicals in this ash that's going to go on to settle in this nearby water body, and then potentially to any communities in the surrounding area. And so, not good, just sort of highlighting that just because something's natural, it doesn't mean that it's good for you. So, on the right hand side here, there are some man made pollutants. And so, if you look closely, you'll see things like plastic bags and maybe some containers. And these are definitely not produced naturally. And so, so humans specifically are definitely a huge source of pollution here in this world. I'm now going to talk about some well-known pollutants or toxicants. So you'll kind of see me use these two words interchangeably. And some examples are listed here. The first one being lead. This is a huge problem around the world, specifically actually in developing countries. And this is a problem primarily because high levels of lead has been associated with renal, reproductive, as well as nervous system toxicity. And specifically, children exposed to lead appear to have really high rates of nervous system problems. The second one on my list here is carbon monoxide. And this is also not very good for you at low levels. People tend to have flu-like symptoms, but at incredibly high levels, carbon monoxide exposure can lead to death. And so a lot of the time, it's a requirement to have carbon monoxide detectors in houses, for example. And then third on this list is this large class of chemicals called endocrine disrupting chemicals, or EDCs. And this is sort of a very broad term for lots of different pharmaceuticals, maybe plasticizers, or just lots of things like pesticides even, which could be endocrine disrupting chemicals. And they're called EDCs because they, the second one on my list interrupt the different hormonal signaling pathways. And so you end up seeing problems with reproduction, growth, or development after exposure to EDCs. EDCs definitely a huge concern for humans, but you also see them being a problem for wildlife. And so this figure here on the left-hand side is a wastewater effluent, and you see the water is really gross. It probably has a bunch of EDCs that's being released into this water body, and now all of the fish and invertebrates in the system are going to have are going to be exposed to these EDCs. And then lastly, I just wanted to touch on plastics. We all know plastics are not good for this world. We try to reduce our plastic use. But what's sort of coming up a lot now is this micro and nanoplastics. And so plastics don't just kind of stick around forever just as they are, but they tend to break into to smaller pieces. And so what you're seeing here is in this figure in the orange circle is some microplastics, so smaller than 100 micrometer, and then some nanoplastics, so incredibly small particles. And what's kind of scary is that they're showing up everywhere in different water bodies, in different plants and food, but we really don't know what the effects are of these particular plastic materials. From here, I'm going to transition into this umbrella term called particulate matter, or PM, which is primarily in air pollution. And so what exactly is particulate matter? It is a mixture of solid particles and liquid droplets in air. The figure on the right-hand side, you're seeing all the haze. And so this is a bunch of particulate matter that's hanging out in the air, and it's obviously not very healthy for you. This PM, particulate matter, consists of several different chemicals. So it's not just one specific chemical, but there's a mixture of different things that's present. And it also encompasses things like dust, dirt, and smoke. If you do a little bit reading about particulate matter, you'll come across terms like PM10 and PM2.5. And the 10 and the 2.5 refer to the size of the particulate matter that's present. And I think this figure on the right-hand side does a pretty nice job of showing what that 10 and 2.5 actually looks like when you compare to human hair. And so human hair is about 50 to 70 micrometers in diameter. And then relative to that, you're looking at a PM10 particle. And then in the bottom here is a PM2.5, which is much smaller and potentially more toxic because these particles are now able to much more easily enter the body and also enter different tissues and organs within the body. And so exposure to PM2.5 particularly has been associated with different respiratory and cardiac system effects. Off note, probably of interest to this audience, if you already don't know this, there was a recent study that came out at the end of last year. And this was a study that was conducted back in 2017. And what they found was that 76.8% of the Indian population was exposed to greater than 40 microgram per meter cube of PM2.5 per year. Well, this number doesn't really mean a lot to you, but I'll tell you that this is actually the limit that's recommended by the National Ambient Air Quality Standards in India. And so the fact that more than 3/4 of the population is being exposed to levels higher than this is definitely concerning. This article also found that 1.24 million deaths in that 2017 year in India were attributed to air pollution. And that's a huge number. And it's something that we probably don't really think about. Because what we're seeing on the other end is people that are affected by different conditions such as lung disease, which includes COPD, lower respiratory infections, and cancer. It could also be heart disease, diabetes, or even neonatal disorders. So lots of different conditions that we see predominantly around sort of in the country. But this study linked several of these back to air pollution, which was really concerning. From here, I would like to transition a little bit to talk about this umbrella term of toxicology. But I'll tell you that this is what you're seeing is that's definition by the National Institute of Environmental and Health Sciences in America. So that's the fact that more than 3/4 of the population what they say is that toxicology higher than this field of science definitely that helps us understand the harmful effects this article also found or situations can have on people, animals, as well as the environment. What's really interesting about toxicology as a field is that it's incredibly interdisciplinary. It encompasses both chemistry as well as biology. And usually, when you think about chemistry, you're thinking about all of the different chemicals that somebody or some animal is being exposed to and how that associates back to the environment as well as different organisms. So, this could be animals or humans or plants even. And when you're talking about biology as a field, you start to see different fields within biology, such as physiology or molecular biology, sort of all falling under this term of toxicology. And I'll just sort of add here, this is sort of one of the reasons why I was interested in becoming a toxicologist is just this idea of working with people, but also just different aspects of science. From here, I'd like to transition into some broad concepts to think about as they relate back to toxicology. And this is sort of the first thing you'll learn if you're in a toxicology class, like the first toxicology class. But what I have here as my first point is the dose makes the poison. And so this was originally, was said by a Swiss scientist back in the 1400s. His name was Paracelsus. And what he said was, all things are poison and nothing is without poison. The dosage alone makes it so a thing is not poison. And so again, just kind of condensed to the dose makes the poison. And just to kind of bring that into perspective, here is a good example. You all are probably familiar with the fruits and vegetables that are here in this picture. And they all have levels of different chemicals that could be not very good for you if ingested at much higher levels than we generally ingest them in. And so just because, again, something is not good for you, it doesn't mean it's going to harm you. Again, it goes back to that idea of taking in a certain amount of dose before you end up seeing effects. Moving on to the second concept that I think is important to bring up, which is that not everyone will respond to a substance the same way. And what this entails is you have to start thinking about things like the amount of exposure or the duration of exposure, both of which will have an impact on the dose. And so if I'm around a chemical a lot, and if I'm also exposed to high levels of chemical, this means I'm going to have a higher dose off that particular chemical on me versus somebody else that's kind of in the other end of the room. And so that is really important when we're talking about how a person would respond. In addition, there's also this aspect of an individual susceptibility. And so we're seeing that other things such as the genetics of a person, age, or lifestyle, which could include things like diet, the microbiome, or even how much you work out, for example, that could have an impact on how much a person is susceptible to a specific chemical or a group of chemicals. I think this graph does a really nice job of showing this. So here on the y-axis, what you're seeing is the number of individuals going from few to many. And then here on the x-axis, we're looking at the response to the same exact dose of some hypothesized chemical. And so you have mild effects here on the left, severe on the right. And this curve shows that majority of the individuals, so many up over here, majority of the individuals have this average response right here in the middle. But there will always be individuals on the left-hand side that are not going to have really severe effects. But then on the other end of this curve, on the right-hand side, you're going to see individuals, think of maybe babies or older people that could have more severe effects. And so really important to think about who is our audience, who are the people that we're studying before we can talk about if a chemical is toxic or not. And then lastly, a couple of fun photos here to kind of show you that there's a difference between hazard and risk. And so this left figure shows a shark, which is considered a hazard because it can potentially cause harm. So think of it as being the chemical. But just because a shark is harmful doesn't mean it's going to go out and eat everybody. What is important to think about is the risk associated with this hazard, the shark, aka the chemical. And so the right-hand side of this figure shows an individual swimming and kind of points to the fact that it's important how close, for example, this individual is to the shark before the risk really increases. I like the picture on the bottom right that depicts this in a nicer, funny way, where this individual on the left-hand side is far away from the shark, not doing anything stupid. So his risk is very low, despite sharks still being incredibly harmful. But on the right-hand side here, you see an individual that's about to jump in the water. And so now the risk goes up. It doesn't mean that the shark is 100% going to attack this individual, but all you can say is your risk has increased. And so now we should start to think about if this chemical or the shark is going to be a problem for this individual. And so hopefully all of that sort of makes sense. From here, I kind of want to transition into this idea of 21st century toxicology, but specifically talking about the challenges that toxicologists are currently facing when in trying to understand effects of different chemicals. And so the first challenge here I have up here is exposure. And so basically, the point that I would like to make here is that it's really challenging to figure out for a individual or a population what they're exposed to, how much of a specific chemical they're exposed to. Are they exposed to one chemical or multiple chemicals? And how do those chemicals interact with each other? And so there's this huge sort of knowledge gap in trying to figure out for lots of chemicals what the exposure looks like for a given individual or a population. The second challenge here entails how there are so many chemicals. And so actually the US EPA estimates that there's greater than 80,000 chemicals used in the US. So if you kind of take into the world, there's probably more than that. But what they say is that most of them have not been tested for both environmental and health effects. And so this is a challenge that toxicologists are currently facing and trying to figure out what is the fastest, easiest, and best way to be able to understand the effects of all of these chemicals. And then lastly, there is this huge problem of mixtures. Chemicals, as you and I know, do not always exist, or they always exist as mixtures. So you're not exposed to a single chemical. And so then questions sort of arise around what is exactly in a mixture. And then also trying to get to the point that mixture toxicity could be really different from individual toxicity. And so a lot of the time you'll see toxicologists studying one chemical at a time, but is that really relevant for a real life situation? And so this is a question that we ask ourselves all the time to try to make it more useful for real world scenarios. From here, I'd like to move on to a case study, which is going to be about this class of chemicals that Ranav had in his introduction for me. And so this class of chemicals is called the polycyclic aromatic hydrocarbons, or PAHs. And this was the subject that I was really interested in throughout my PhD. And so I thought I'll just kind of show you the types of things that not only I've gotten to do, but also just the field is working on currently. So this slide has different sources of PAHs. PAHs are found are produced in forest fires. They're also in volcanic eruption. So this nasty volcanic eruption definitely has tons of PAHs here in the dust and the ash that's being produced. Oil spills actually are a huge source of PAH exposure, and they're in fact the largest source of PAH exposure in the oceans. And PAHs end up actually on the surface of the water here, and then they can kind of get into different animals and have negative impacts on these animals. Cigarette smoke is an important source of PAHs, primarily for the people smoking, but we're also seeing lots of research on secondhand smoking and even third-hand smoking now. And then on the right-hand side here, there's vehicle exhaust. So just exhaust out of vehicles is a huge source of PAHs. And then something that we might not think about if you're out grilling food, that char on the food is actually contains some PAHs. And so if you're touching it, now you have some dermal exposure. If you're eating the food, now you're getting some PAHs into your body. And so lots of different sources, but then the big question is, well, what the hell are PAHs? They are a class of common environmental contaminants. They are products of incomplete combustion. What this means is when you're having a burning reaction, we all know that we need oxygen for a burning reaction to take place. But if there's not enough oxygen present, or the reaction's going on too quickly, then you end up creating these large chemicals called PAHs. They are a class of over 1,000 chemicals. So it's not just one or two, but there's over 1,000 of these chemicals. And they're characterized by these benzene rings. And so this is an example of a benzene ring. And so you'll see that there's multiple benzene rings all kind of fused together. And then that's sort of characteristic of a PAH. So what are the health effects of PAHs? They are well-known mutagens and carcinogens, which means their exposure has been correlated with different types of cancers, specifically skin and lung cancer, as well as bladder and gastrointestinal cancers. Exposure to PAHs has also shown to decrease pulmonary function, also correlated with cardiovascular toxicity as well as immune system toxicity. And so this is kind of very relevant to my thesis, but also sort of relevant to what toxicologists within this field are currently interested in, because new research is showing that lots of new correlation research is showing that exposure to PAHs decreases birth weight and length, could also decrease head circumference, and also increase childhood obesity rates. In addition, there's also some neurological effects where, again, that exposure to PAHs is associated with a decrease in IQ as well as an increase in ADHD rates. When you think about this from a laboratory perspective, scientists like to use different model organisms, so typically rodent models like mice or rats. And when these animals are exposed to PAHs, we find that many of the PAHs are indeed developmental toxicants, where in the lab, they end up causing cardiovascular toxicity, oxidative stress, as well as persistent neurodevelopmental effects. And so just that early exposure can lead to long-term neurodevelopmental effects. And so now I want to kind of bring back the slide on the 21st century toxicology challenges, but sort of just link it back to PAHs. And so the first one being exposure. So this holds true for PAHs, where we really do not know what the exposure levels are for different populations around the country or world even, and what sorts of chemicals are in different PAH, which is umbrella term for thousands of chemicals, right? And so what specific PAHs are present in these mixtures that people are being exposed to. Also, as I pointed out, there's over 1,000 PAHs, and most of them have not been tested for environmental and health effects. And so, again, another challenge that's present in the field. And then lastly, like, as I hinted at before, PAHs are always existing as mixtures. And so this means that until we get a sense of the types of mixtures that are present, depending on the situation and what's in them, we won't really get a good idea of the different toxicity effects that are caused after exposure to these chemicals. So just to tackle exposure, this is something that I have personally not worked on, but I thought was important to bring up. This is a team in my graduate school, so at Oregon State University. There's Dr. Holly Dixon, a friend and colleague on the left-hand side, and here's Dr. Kim Anderson, who pioneered this idea of using these silicone bands. You might have used these bands sort of as kids. I've used them in school just for fun. But what she thought about was actually using these bands as a way to sample PAHs. And so what she does is she makes these bands, puts them on different people for a certain period of time, and then takes the bands back to the lab and then looks to see what sorts of PAHs are present within these bands. And so that'll kind of give you a sense of what the exposure was like for the person that was wearing the band. And so from there, what you can do is now you can say, okay, this person was exposed to these chemicals. Now let's take these chemicals and try to figure out, are they good or bad? Are they harmful, toxic? And so how do you go about doing that? You can essentially, you could use different model organisms. And so I pointed out before that typically toxicologists tend to use different rodent models like mice and rats. But here I want to introduce to you a model that I got a chance to work with throughout my PhD, which is the zebrafish model. And so, what exactly is a zebrafish? And so, here's a photo of an adult zebrafish. You can see immediately why it's called a zebrafish. And so, I guess a bit of trivia is that these fish are actually tropical fish from India. And so, all of those red dots are telling you where exactly these fish are found in India. So, sort of in this northern region, also including countries like Nepal and Bhutan over here. And so scientists actually realized that this could be a really good model to use for both toxicology, but also just lots of developmental studies. So why exactly do we do this? Zebrafish have transparent embryos, and they also develop very rapidly and externally. And so as a scientist, we're able to put these zebrafish embryos under a microscope and visualize their development. What you're seeing here is a zebrafish going from a single cell stage all the way to 24 HPF, which is hours post fertilization. And so, in just that 24 hours, it goes from that single cell to an organism with a head and a tail. And by the time it's 120 HPF, so seen here on the right hand side, which is 5 days, they're actually fully functioning and they have a well developed brain. They have a heart, a cardiovascular system, a liver. And so they're able to conduct all of these different functions in just 5 days. Additionally, zebrafish are also amenable to whole animal mechanistic evaluations. And so what this means is I'm going to kind of, to help you out, I'm going to compare zebrafish to a whole animal rodent model. So this is what's typically used. However, it's important to point out that rodents have a much larger, longer lifespan, and they also only produce fewer number of offspring. And so maybe rodents will have 2 to 3 offspring versus a pair of zebrafish can have about 200 embryos, which means that we have a large enough sample size and we're still able to kind of maintain that whole animal approach. Toxicologists will also use cells in a dish, so a cell culture system, and expose cell culture to different chemicals. However, you lose out on this whole animal approach. And so we're to try to find kind of a balance between this in vitro cell culture system and this rodent model. Scientists have been proposing to use zebrafish as a sort of an in-between to help measure the effects of different chemicals in a quick way in a whole animal system and also have some mechanistic insight. So hopefully that made sense. But from here, my last point about zebrafish is that they actually have a pretty high genetic similarity with humans. So this was a study that was published in 2013 that sequenced the zebrafish genome. And what they found was that approximately 70% of human genes actually have at least one obvious zebrafish ortholog. And so this means that if you're using zebrafish as a proxy for humans and you identify certain genes within the fish that are important for toxicity of a chemical, and humans have this gene, you can now use the zebrafish model to understand this particular gene and what its role is in toxicity. I thought I would also kind of give you some insight into what a zebrafish facility looks like. So the picture here on the left is actually one of the rooms in the lab that I got a chance to work at. So you're seeing multiple racks, and zooming in onto one of these rows, for example, you're seeing individual tanks with lots of different fish, and you're noticing probably all the different colored labels. And so typically scientists will have fish that are exposed maybe to different chemicals. They might have some mutations. We've gotten a chance to use techniques like CRISPR-Cas9 on zebrafish and then grow them out in different tanks. And so that's kind of what you're looking at over here. And these are all adult fish. And so far, I've been kind of going after this idea of developmental toxicity. And so it's important to go after the embryos that these zebrafish make. And so one way to do that is we set up fish in these spawning tanks. And so here there's a barrier and with the adult fish on the top, and the embryos are present in the bottom. And so the adult fish are not able to eat the embryos, which is what they generally do. And with the barrier, they're not able to eat them. And so now scientists can collect these embryos and use them for different studies. Specifically for PAHs, but also holds true for other chemicals. I wanted to show you what the exposure paradigm is in developing zebrafish. This is only one way to do it. It's the way that we did it in our lab, but you'll see sort of versions of this across the board in different laboratories. But what you're looking at here is a zebrafish developmental timeline going from 10 minutes post-fertilization, so that one cell stage, which is what the little bump is over here, and then going through development all the way to 120 HPF, which is 5 days. What we do is, as soon as the embryos are laid, we collect them in a strainer of sorts. And here you'll see at the bottom is actually a pool of maybe 2,000 to 3,000 embryos that are at the bottom that we can collect. Another zoomed in image of this, of a zebrafish embryo. And you see that they actually have a chorion, so sort of like a shell around the embryo. And sometimes you can remove the chorion, and other times you can leave it on before you expose these fish to chemicals. And then we expose zebrafish at 6 HPF. So 6 hours later, we expose fish to a concentration range of PAHs. And so getting to that idea of dose, so you don't want to only expose to one particular concentration. So we kind of want to go low. We also want to go really high to get a sense of what the different effects are at these different concentrations. We actually use these 96 well plates. And what we can do here is we can place an individual fish in an individual well of this plate. And so also getting to the concept of susceptibility. And so we can ask questions about if a fish in one well, is it going to respond differently from a fish in another well, both of them being exposed to the same chemical at the same concentration. And so we like to do studies like that because we have this handy tool of a 96 well plate. At 24 HPF, so this is 1 day after these embryos are born, they have a head and a tail, remember, we can conduct a behavior assay. And so here's just an example of one of these. And I'll just point out this graph at the bottom. What you're looking at is movement on this y-axis, and on the x-axis is time. And so what we do is we put the zebrafish in this box, we shut them in there, and it's all dark. And so they're just kind of hanging out, seen here in the graph. But then this particular red spot, or this light here, essentially is actually zebrafish being exposed to a flash of light. And as soon as they're exposed, they actually kind of go crazy. So they start to move around a lot. And so you see this increase in movement, and then it settles down. And then we give them another pulse of light. And this time they actually do not respond. So it's almost as if they acclimated to the light and they're not going to freak out the second time around. And so we can do this for normal fish, but we can also do this for fish that are exposed to chemicals. And so any deviance from this normal behavior will tell you that the chemical actually impacted something that has to do with this particular behavior endpoint. At 120 HPF, so this is at 5 days, we can once again conduct another behavior assay. And this is very similar to what you saw in the previous slide, except this time we're alternating between periods of light and dark. At this life stage, zebrafish do not move a lot in the light, but then in the dark, their movement increases, and you kind of see this cycle through. And once again, any deviance from this normal behavior. So, for example, if you kind of focus in on this side, you see that the red is a lot higher than the black, which is the normal or the control. And so now you can say that, okay, this particular chemical actually altered how the zebrafish behave in this particular test. We can also look at morphology. And so this is what a normal zebrafish looks like. But upon exposure to chemicals, you start to see different malformations that form. And so this is an edema malformation, which is essentially a fluid-filled region around the heart. You could also have a bent axis, malformations to the jaw and the snout, as well as a really severe caudal fin malformation. And so looking at this, we can kind of get a sense of which PAHs are causing what effects, and how is this really toxic for the zebrafish. And then lastly, something that's kind of pretty new in the field, we can also conduct RNA sequencing. And so what we do is, after exposure to either an individual pH, you could also expose fish to a mixture of chemicals and a control. We conduct RNA sequencing. What exactly is RNA sequencing? This is what it'll do is it'll give you an unbiased snapshot of gene expression for the PAHs in the whole animal. And so just kind of taking you back to biology 101, here's DNA. DNA goes through a process called transcription to make RNA. We're all familiar now with this idea of RNA from the mRNA vaccines, for example, for COVID. And so this particular technique, what it'll do is it'll tell you what are all of the different RNA molecules that are present in an organism at a specific time. And so this is telling you which genes, so the DNA, are important because they are being made into RNA and then potentially into protein later. And so this tool is really cool. We can use it to identify essentially which genes are important because they're being transcribed into RNA. And so now just kind of linking back zebrafish specifically for 21st century toxicology challenges. Using this model, we can begin to understand which PAHs are harmful. We can also try to figure out what exactly are these harmful effects. And so this is by running lots of different tests, such as those morphology tests, as well as behavior tests, and other labs and us also will do kind of other things depending on the chemical, but this is pretty standard for PAHs. You could also ask questions like, how do the PAHs cause harm? And so that RNA sequencing is key here because it tells you which genes are important for toxicity. And once you identify specific genes that are important within zebrafish, you will start to ask questions about, well, are these genes present in humans? And what does that mean for humans? And then you can kind of ask follow-up questions, conduct more experiments to get a sense of which genes are actually important. And then you can also try to figure out what the mixture effects are. And so here I kind of showed you generally what we do for individual PAHs, but you can kind of follow through and do similar sorts of studies using mixtures of different PAHs and then kind of do comparisons. And then just to reiterate these important points in that one, using zebrafish as a model organism is a lot faster than rodent models. Again, going back to how quick development is, you can just kind of conduct many of the studies in just 5 days and use this really sensitive model to understand effects of different chemicals. Also, again, reiterating a previous point that this is a whole animal approach, and this is different from commonly conducted cell culture studies where you cannot take into account things like metabolism of the chemical. And so it's really important to, it's a really important pro of using the zebrafish model. And then lastly, I think this is sort of a really important point, especially for all of us animal lovers out here. The ultimate goal is to create a database with toxicity information from these zebrafish studies to mine for predictive toxicology. And what this means is that hopefully within your life and my life, we will have maybe a website, a software where you can go in and enter in any chemical, new or old, and try to figure out what the potential toxicity effects are of that chemical based on all of the other data that's already been collected from zebrafish. And so, in theory, you wouldn't have to use any animals in the future. And so, that's what we're all hoping for. But we got to start somewhere. And so, this is kind of a good tool that's becoming more and more common, especially now. And then to sort of finish up this presentation, I thought I'd just wrap up with a few key takeaways. The first one being that several chemicals are present in our environment whose toxicity effects we know absolutely nothing about. There's also a high correlation between pollution and the negative health effects in both humans and also in other animals. And this is something I didn't talk about over here, but there's plenty of evidence showing that. And then I hope I convinced you that there is a huge need to gain a better understanding of what exactly we're exposed to. And then finally, zebrafish is a helpful tool to understand the toxicity of lots of different harmful substances. With that, I will be happy to take any questions. I put my email on here. I am pretty responsive to email, so if you have any questions, feel free to shoot me an email and we can have some really interesting discussions. With that, I'll hand it over to you guys, Rana and everybody else.
Well, I think I have two questions. I'll begin with the first one and then you can answer, and then we'll move on to the second question. I think my first question is regarding the exposure of toxic chemicals. Now, we cannot control our exposure to toxic chemicals. That's actually beyond our reach. So, how do you think that we know which chemicals that we are exposed to, and do we control their negative harmful effects on our own health? Because you mentioned in the last slide that there is there is a negative effect of exposure to chemicals and the effects on health.
Okay, yeah, that's a good question, and I think there's several parts to that, so I'll try to answer kind of different parts to that. But just sort of thinking about like your lifestyle and my lifestyle. Yeah, absolutely. Like you and I do not know what we're being exposed to every single day. Can we control this? Well, yes and no, right? So I think it's important to kind of just be in the know of what sorts of chemicals are around us, in terms of like from studies saying that, okay, if you go in this particular area, you're going to be exposed to high levels of lead, so stay away from there. Lead is not good for you, do not go by the lead. And so if you have that prior knowledge, I think you can think about what you would like to reduce in your life. But that being said, I mean, if I have some shampoo and I know that it's really bad, but it's the only shampoo that works for my hair, I can't put it away, right? I'm going to have to use it. And so it's just sort of balancing those things and like trying to figure out, if I use a shampoo, okay, so maybe I'll, remove the soap from my life or something like that. So kind of just balancing different things um, to try to make the best possible decision in this moment of time. Because again, once again, remember, we don't understand the effects of majority of the chemicals, and we're just starting to get there. And so it might be too premature to make those sorts of decisions, but just be very, the news is okay, the media does a decent job, but if we like to read papers, then go ahead, find those papers to really figure out if a chemical, what we know about a chemical, and then go from there. Hopefully that answered your question.
And just a follow-up question to that: do you think that there are any public policy systems that we can put into place to sort of have a larger governance model where entire populations are not exposed to a certain kind of chemical, like with the toxicity that we already know? Is that possible?
Yeah, so I think it would really depend on the chemical, but for sure, I mean, there has been, for example, so the one good example I can think about is bisphenol A. So this is a chemical that's present in plastics, or that used to be present in plastics, and what we started to see was that like babies from their bottles were being exposed to bisphenol A, and then there were studies showing the negative health effects, and so now essentially the US has banned bisphenol A in any plastics specific to babies. And so yeah, so there is policies that are being made, that have been made in the past, not just for bisphenol A but for other chemicals, after we have certain amount of information about the toxicity of them. There's also, I guess I'll just stop there. I mean, I think that probably answered your question.
That it. And the second question coming to you, you spoke about gene toxicity and even the toxicity within the genes and the sort of correlation that is there, or say the similarity would be a better word between the genome of the zebrafish and the humans, like approximately 70%. Now, I was recently reading this research paper where where researchers have identified toxic genes in E. coli genome, and the overdrive of certain proteins have actually caused toxic expression, because you also talked about gene expression and RNA sequencing. Even most of my research with my extended essay is based on gene expression and RNA sequencing. So just connecting back to that, is there any toxic genes, so as to say, to use the specific terminology, identified in zebrafish, and what does that actually mean for humans because of the high percentage similarity of the genomes?
So I would kind of I'll try to unwrap that question, but I'm not familiar with any specific gene that's sort of toxic in the grand scheme of things. However, depending on why a particular gene is turned on to be expressed, could kind of have an effect on the other end. So, for example, you have a chemical that a zebrafish is being exposed to. It is going to turn on a bunch of genes. Now, what if one of the genes that it turns on actually goes back to the chemical and changes the chemical to make it more toxic, to be more harmful to the zebrafish? So this can be pretty bad. And so I think it would be more context dependent, where a gene could end up being not very good, even though it's being expressed. But I can't think of sort of the E. coli correlation in zebrafish.
Ranav, do we have any questions from our audience or something like that? Then I think we can close. It was a very informative and a very thought provoking presentation, Dr. Shankar. Thank you very much for that, and I hope even our audience, I'm very sure that all three of us did enjoy. And we had a lot of takeaways from that, and it sort of allowed us to reflect on our own readings and our own environment, so as to speak. So thank you very much for that, and hope to stay in touch and interact on an even more, and sort of, because we're all applying to US universities next year, hope to meet you in person as well and take this discussion forward then.
Yeah, absolutely. Thank you so much for inviting me. This was really fun. And I mean, this is kind of cool. I mean, I was talking to Rana